Okay, good afternoon everyone. It's James Walters here from Cardiff University. I'm the co-chair of the PGC Schizophrenia Working Group. Myself, Pat and Jerome Breen have been thinking about developing the worldwide lab meetings, and the model we've gone for, as you may know, is that there are some stellar senior PIs who get hour-long talks, and Josh Gordon and others have given those recently, but we also wanted to encourage earlier career researchers to be involved and share their work with the community. And so what we thought was a good way of doing that was to have 3 or 4 shorter talks for the Worldwide Lab meeting. And today we've got 3 individuals, each of whom are postdoctoral researchers here at the MRC Centre for Neuropsychiatric Genetics and Genomics in Cardiff. Sophie Legge, Elliott, Rees and Alex Richards, who each are going to be talking to us today about research which is being submitted, under submission, or in the process of being published. So hopefully is of interest because it's pretty current, and we'd really encourage people to ask questions. And the way to do that is that at the top of your screen, you can see mine being shared now. You should have a Q&A box. If you click on that, then you can ask questions. So do that through the individual talks. The other option is to go and via the chat, is to ask questions via chat. And so at the end of each talk, we'll then pull up the Q&A and chat questions, and then the researchers will be able to address those questions. And we hope each talk is going to be about 15 minutes with 5 minutes for questions at the end of each talk, and we should be done in time for within the hour. So thanks very much, and without any further to do, I'm going to hand over to Sophie Legge.
Hi, so as James said, I'm Sophie Legge, one of the research associates here in Cardiff, and I'm going to talk to you today about a genetic association study of psychotic experiences in the UK Biobank. So psychotic experiences such as hallucinations and delusions are features of psychiatric disorders such as schizophrenia and bipolar disorder, but they're also reported by around 5 to 10% of the general population. Now, they're only considered to be symptoms of psychiatric illness if they co-occur with other features of that disorder, including some aspect of impairment. Heritability estimates have ranged from around 30 to 50% from twin studies and from 3 to 17% from SNP-based studies, and this suggests that genetic variants may play a role. It's currently unclear whether subclinical psychotic experiences should be thought of as either 1, on a spectrum that at the extreme end specifically relates to schizophrenia, 2, are largely unrelated to the psychotic experiences experienced in schizophrenia and other major mental disorders, or 3, they're related to a liability to general mental disorders. And epidemiological evidence currently supports that third hypothesis. So psychotic experiences in adolescents have been associated with a range of disorders in later life, including schizophrenia, but also depression, self-harm, and PTSD. Although there have been 3 genome-wide association studies to date, we haven't identified any genome-wide significant loci, likely due to power considerations, and so we're yet to show this hypothesis from a genetic liability perspective. So our aims were to use a UK Biobank sample to identify genetic loci associated with psychotic experiences in a subclinical adult population, and to determine whether genetic liability to psychotic experiences is shared with schizophrenia and or other psychiatric disorders. And we did this in 3 ways: by looking at genetic correlations from the genome-wide association study from part 1, looking at polygenic risk scores and copy number variation. So as I'm sure you're all aware, the UK Biobank is a population-based cohort study of around 500,000 individuals aged between 40 and 69. The psychotic experience phenotypes we used came from a mental health questionnaire that was emailed to participants in 2017, and it was completed by just over 157,000 individuals. As part of this questionnaire, everyone was asked about various symptoms relating to psychiatric disorders, and they were also asked about their lifetime history of psychotic and unusual symptoms. So we considered 3 different phenotypes. The first, which we call any psychotic experience, is classed as a positive response to any of the 4 questions, and they were regarding visual hallucinations, auditory hallucinations, delusions of reference, or delusions of persecution. The second phenotype was a subset of this first group, as you can see from the Venn diagram down here, which describes the overlap. So we took those that reported any psychotic experience, but they also rated it as being a bit, quite, or very distressing. The third phenotype that we used is another subset, but this time we're only including those where the experience occurred on more than one occasion, so they're having repeated psychotic experiences. Around 121,000 individuals gave a negative response to all 4 of those psychotic experience questions, and these comprised our comparator group. So we want to exclude individuals with schizophrenia, bipolar disorder, or psychotic disorder from the analysis to ensure that any associations weren't driven by these individuals. And to do this, we tried to identify them from all available sources that we had. So that includes self-reporting at the interview, during the mental health questionnaire, hospital records, and death records. And this was 1,300 individuals that were excluded from all further analyses. In terms of method, the genetic data we used was provided by UK Biobank, and we applied some additional filters to ensure we had high quality SNPs. 1 member from each related pair with a kinship coefficient above 0.15 was excluded. We preferentially retained those individuals that reported a psychotic experience, and otherwise we removed them from random. We also excluded those that weren't from a European genetic ancestry, and we defined that as self-report or using the principal components. So we conducted 3 genome-wide association studies, 1 for each of those phenotypes I described in the previous slide. And we used SNP test and the BGen imputed data. We covaried for principal components and the genotyping array. We used FUMA for functional annotation, and to calculate heritability on the observed scale, we used the LDSC. Now, we wanted to try and validate this genome-wide association study, so we applied the summary statistics as a polygenic risk score in the ALSPAC cohort. So moving on to the methods now to look at the overlap, potential overlap with other psychiatric conditions. We use genetic correlations using LDSC, and we calculated them with schizophrenia, bipolar disorder, major depression, neuroticism, intelligence, ADHD, autism, and the PGC cross disorder. To create polygenic risk scores, we use polygenic risk scores from schizophrenia, bipolar depression, neuroticism, and intelligence, and using the method described by the PGC at 11 p-value thresholds. Now, the intelligence GRAS was actually the only sample that deduplicated at the genotype level with UK Biobank, and so all these other findings should be treated with caution. And our primary reason for using them was to investigate these additional phenotypes that weren't powered enough for LD score regression. The methods of the CMV calling has been previously described by members of our group, and we compared carrier status for rare CMVs previously associated with schizophrenia and neurodevelopmental disorders. So moving on to some results. On the right-hand side of your screen, you can see we have a Manhattan plot for each of these 3 genome-wide association studies. And in this table are the variants, the independent variants that survived correction for multiple testing, so those that passed the genome-wide significant threshold. So as you can see, we identified 4 loci, 1 of which intronic to ANK3 that was associated with any psychotic experience, 1 intronic to CNR2 that was associated with distressing psychotic experiences, and 2 intergenic loci. Now, the heritability estimate was low for the any psychotic experience GRAS, and that was estimated at 1.71%. Now, unfortunately, the other 2 GRAS didn't meet requirements for LDSC, given that their case number was below 5,000 in the cases. So I just wanted to say a few words about these 2 loci. So we know that ANK3 is one of the most replicated genes for bipolar disorder, and variants in this gene have also been associated in the PGC cross disorder GRAS and in a rare variant analysis of autism. And CNR2 encodes for CB2, which is a cannabinoid receptor. And that's interesting given we know that the cannabinoid system has been implicated in schizophrenia and depression. So given the association of CNR2, we tested but found no evidence of a mediating or moderating effect of cannabis use. So although cannabis use was significantly associated with psychotic experiences, as you can see here, the SNP we identified was not associated with cannabis use. Furthermore, when we add cannabis use as a covariate or an interaction term, the relationship between the CNR2 SNP and psychotic experiences remain. That said, we don't believe that this necessarily means that we can rule out an effect of cannabis use, given the limitations of the self-report measure in UK Biobank on substance use. So taking these summary statistics from any psychotic experience GWAS, we created a polygenic risk score in ALSPAC to see if it would predict psychotic experiences in another sample. Now, this polygenic risk score was weakly associated with psychotic experiences at age 12 and 18 at thresholds below p-value 0.05, which is, you can see it in the graph down here. But this polygenic risk score was also associated with major depressive disorder at 18 as well, and perhaps actually more significantly associated. So moving on now to look at the shared etiology with neuropsychiatric conditions, the genetic correlation analyses showed that the psychotic experiences GWAS had a significant genetic correlation with schizophrenia, depression, ADHD, autism, and the cross disorder GWAS. So we're looking down here. So the strength of the blue is how positive the genetic correlation is, and the stars represent significance. So interestingly, we did identify a relationship with schizophrenia, but actually that was weaker than some of these other neuropsychiatric conditions. Unfortunately, as I said earlier, the other psychotic experience phenotypes weren't powered enough for the genetic correlation analysis, so we could only do this for the larger any psychotic experience analysis. So as I mentioned earlier, the polygenic risk score results are caveated by the fact that the training sets haven't been entirely deduplicated from UK Biobank. Now in this plot here, down the y-axis you can see the different polygenic risk scores, and on the x-axis is the odd ratio, and here we can see the effects for any psychotic experience, and now we can see the effects for those multiple occurrence experiences and distressing experiences. So we did find that there were significant associations for genetic liability with schizophrenia, bipolar disorder, and depression. And interestingly, these associations appeared to be stronger for those distressing experiences. We also applied polygenic risk scores to look at those 4 individual symptoms that we asked. So we asked about visual hallucinations, auditory hallucinations, delusions of reference, and delusions of persecution. And interestingly, we can see that this effect appears to be stronger for delusions, and in particular for delusions of persecution. So that said, we'd like to make it very clear that the variance explained here is very small. So to give you an example, these 4 circles are perhaps the most significantly associated results, and the R squared for all of them is below 1%. So although they are associated, it's certainly not explaining a large amount of the variance. So lastly, we found that those reporting psychotic experiences were more likely to have a rare CMV that was previously associated with schizophrenia and neurodevelopmental disorder CNVs. And again, interestingly, this association appears to be stronger for distressing experiences, increasing, going up to an odds ratio of around 2. So before I conclude, just to briefly outline some of the limitations of this study. So although we used all the available information to identify and remove individuals with a psychotic disorder, it's likely that there may be some individuals not identified and remained in the analysis. A further limitation is the use of a retrospective measurement of a lifetime psychotic experience. And there's evidence of healthy volunteer bias in UK Biobank. And indeed, we found that participants recruited and that those that fulfilled their MHQ had an increased polygenic risk score and a decreased schizophrenia, depression, and neuroticism polygenic risk score. And lastly, the polygenic risk score training sets weren't deduplicated at the genotype level. So in conclusion, we conducted the largest GWAS to date of subclinical psychotic experiences in UK Biobank, and we identified genetic loci in variants intronic to ANK3 and CNR2. And interestingly, these appear to be genes that are implicated across several neuropsychiatric disorders. And we feel that the main finding really of this work is that although psychotic experiences were found to share genetic liability with schizophrenia, they also share genetic liability with depression and neuropsychiatric and neurodevelopmental disorders. And this was consistent across our 3 analyses of correlations, polygenic risk scores, and CMVs. Furthermore, those distressing psychotic experiences seem to be indexing more of a mental health liability. Lastly, given how low the heritability estimate was and that the total variance explained by the polygenic risk score, it's unlikely that the genetics of subclinical psychotic experience will have an important impact in understanding the genetics of schizophrenia or any other mental health disorder specifically. And it's certainly not in a specific way to schizophrenia. I'll do the last slide. Okay, and I'd just like to acknowledge all my colleagues at Cardiff and some of our collaborators from Bristol, King's College, and in Amsterdam, and thanks to the participants of UK Biobank and ALSPAC.
Excellent, thanks very much, Sophie. So I'll just stop sharing there and have a look if there's any questions. Okay, there don't appear to be any questions at the moment. Please, just as a reminder, if people have got questions, then there's a Q&A folder on the Zoom outline, or they can do it via chat. But at the moment, there's nothing there. So if people have got questions relating to Sophie's talk, then we can also take those at the end. But in the meantime, we'll switch over to our next speaker, who's just preparing his talk. So next is Elliott Rees, who again is a postdoctoral researcher with the Schizophrenia group in Cardiff.
Right.
Okay.
Share.
Yeah. Just. I'd go to the desktop.
Oh, is it?
Oh no, that is that. It's got the green rings again.
Okay, so hopefully you should be able to see those slides.
We're relying on that, aren't we?
Yeah, please shout if you can't.
Okay.
Hi, so I'm Elliott Rees. I'm a postdoc at the Schizophrenia Research Group at Cardiff University. And I'm going to talk about some recent work that we have as a preprint at the moment on bioRxiv, looking at both de novo and common alleles in a schizophrenia proband parent trio sample. So we know that schizophrenia is a highly polygenic disorder with important contributions from both rare and common alleles. And we know that common alleles with an odds ratio of less than about 1.3 contribute to at least a third of the liability of the disorder. And we also know of a number of rare copy number variants, between about 8 or 12, that are robustly associated with schizophrenia. And we know of one gene, SETD1A, that's enriched for ultra-rare loss-of-function variants discovered through exome sequencing. And a lot of these rare variants are known to arise as de novo mutations, so they haven't been inherited from the parents. They're new mutations that have occurred in the gametes of one of the parents. And we know that de novo mutations are important risk factors for a number of different neuropsychiatric disorders. So, for example, so taking this from a recent preprint by Dan Harrigan. So exome sequencing studies have shown that loss-function de novo variants, you see a greater excess of these mutations at an exome-wide level compared to the mutation expectations. So we see a subtle but significant excess of loss of function de novos in schizophrenia, and an even greater excess of them in earlier onset neurodevelopmental disorders such as autism, developmental delay, or intellectual disability. But we know that these de novo mutations are not randomly dispersed across the exome. They're largely concentrated within a relatively small subset of genes, and we know that they're concentrated within around 3,500 genes that are known to be intolerant to loss of function mutations. So you generally don't see individuals with loss of function mutations in healthy controlled populations. And the de novo study design has been particularly effective at identifying individual genes associated with early onset neurodevelopmental disorders. So for example, there's 94 genes associated with de novo variants in developmental disorders, and there's 99 genes associated with autism spectrum disorder. And some of these genes are shared, so there's about 33 genes that are shared across these disorders. But to date, there's only one gene in schizophrenia, SETD1A, that's associated with rare de novos. And there's two reasons for this. One is that, as you can see, de novo mutations don't contribute as much to the liability of schizophrenia as they do for some of the earlier onset neurodevelopmental disorders. And the second important point is that the sample size, or the number of trios that have been analyzed for de novo variants of schizophrenia, is much smaller than in autism or in developmental delay. Which brings us on to one of the main aims of the current studies, which is to analyze de novo variants in a new sample that we've produced here at Cardiff, a new schizophrenia sample of 613 trios, and then to combine this with published data for a total sample size of 3,444 trios, which is the largest analysis of de novo variants in schizophrenia to date. And our two primary aims were to try and refine the sets of genes that are enriched for de novo variants in schizophrenia, and to identify novel genes that are enriched for de novo variants. So to briefly describe the new sample, so we produced exome sequence data here at Cardiff on Illumina HiSeq 4000 instruments, sequencing probands diagnosed with schizophrenia and both parents. And these trios were recruited from various sites across Europe. And we sequenced 2,000 individuals from 674 trios before QC. And then after performing standard sequencing quality control, we were left with 613 new trios, which we could call de novo mutations. So we called de novos using in-house scripts. And then to make sure that we had a high quality call set, we performed two analyses. The first was to look at the rate of de novo mutations in our new trios and compare it to previously published data sets. And what we found is that in the 630 new trios, we observed 606 coding de novo mutations. So on average, about one coding de novo per trio, which is in line with previous expectation rates. And this coding de novo rate was not significantly different from that previously observed in around 2,800 trios. And we also found that the de novo mutation rate in our new sample didn't differ to previous schizophrenia trios when we broke it down by mutation annotations, so for loss of function, missense, or synonymous de novos. So this indicated that we weren't grossly undercalling de novo mutations in our new sample, or we weren't grossly overcalling them as a result of calling false positive mutations. But we also wanted to perform a validation experiment. So we used Sanger sequencing to independently validate a subset of de novo mutations. So we targeted all loss of function de novos in our new sample and a random subset of additional missense and synonymous mutations. And when we did this, we found very high validation rates. So 95% of the point mutations validated and all of the indels. So moving on to the gene set analysis, we first wanted to examine the enrichment of loss of function de novo variants in two broad categories of genes. So the first being genes that are known to tolerate loss of function mutations. So our loss of function tolerant genes, of which is about 15,000. And we use this as a negative control because previous work has shown that there's no evidence for an enrichment of de novo variants within these genes in schizophrenia. And that's what we observed in our new samples. We observed in our new trios no enrichment of loss of function de novos in these loss of function tolerant genes, indicating that our analysis was well calibrated. But when looking at loss of function intolerant genes, we saw a significant enrichment of loss of function de novos in both our new trios, as expected in the previously published literature, and then a highly significant excess in a combined analysis of all trio data. We also wanted to look at those 160 genes that have been previously associated with de novo variants in early onset neurodevelopmental disorders, given the wealth of data showing that there's a shared liability across these neuropsychiatric phenotypes. And when we looked at this 160 genes, we saw evidence that there was an enrichment of loss of function de novos in our new sample. It wasn't significant, but this is a relatively small number of genes, so it's going to be underpowered. But a very strong enrichment in the previously published trios, and very strong enrichment, of course, in the meta-analysis. And in fact, the enrichment of loss of function de novos in these new developmental disorder genes was significantly greater than the enrichment observed across all loss of function intolerant genes. So moving on to the individual gene analysis. At a genome-wide level, after correcting for all genes tested, we didn't observe any single gene to be enriched for de novo mutations after correction for multiple testing. But we wanted to do a focus analysis on those 160 neurodevelopmental disorder genes, because previous work, some from our group, has shown that by focusing on mutations or genes that are associated with, say, intellectual disability is an effective way for identifying novel schizophrenia risk factors. So looking at these individual 160 genes, we tested for an enrichment of loss of function variants, of damaging missense de novo, so this was defined using the MPC score, and a combined analysis of loss of function and the damaging missense. And when we did this, we observed one gene to be enriched for missense mutations, missense de novos, after correcting for multiple testing. And this gene is SLC6A1, also known as GAT1, which is a gamma aminobutyric acid transporter. So its main function is to remove GABA, the main inhibitory neurotransmitter, from the synaptic cleft. So we observed 3 missense damaging mutations in this gene, 2 of which were in our new trios, and 1 was in a previously published trio. And of course, on the basis of this analysis, this is a known neurodevelopmental disorder gene, and it's associated with both autism, developmental delay, and epilepsy. So here I'm showing a figure from the recent autism sequencing study by Satterstrom, which came out last December, I think, on bioRxiv. And this figure shows the genes associated with rare variants in autism and the number of times they're disrupted by missense variants on the Y, and the number of times they're disrupted by loss of function variants on the X-axis. And they highlighted in this paper that there was only 4 genes that were preferentially disrupted by missense variants compared to loss of function variants. And as you can see, one of those 4 genes is our new candidate risk gene, SLC6A1. So in autism, this is pretty much an exclusive enrichment from missense variants in this gene. And we also see an enrichment of missense variants in this gene, which adds to the biological plausibility of our finding. So on the basis of that, we decided to meta-analyze all the schizophrenia data and all the autism data for this gene. So when looking at autism on its own, missense variants are associated with SLC6A1 with a p-value of 8 times 10 to the minus 11. But when we added the schizophrenia data to this analysis, the strength of association increased by several orders of magnitude to 10 to the minus 14. But the picture is slightly different in epilepsy and in developmental delay. So if, for example, in epilepsy, loss of function mutations contribute to at least to around 50% of the de novo mutations observed in this gene. It's around 30% in developmental delay. And I've taken these numbers from the Satterstrom paper. So moving on to the second main aim of our work, in which we wanted to investigate the relationship between de novo mutations, in particular risk de novo mutations, and common risk alleles for schizophrenia. So to do this, we couldn't rely on the exome sequencing data by itself. So we had to combine the exome sequencing with SNP genotype data, which was available in 1,122 of our sequenced trios. So as a brief background as to how we analyze common variants in a trio sample, because it's slightly different to the case control study designs. So a couple of years ago, a new method came out for this purpose by Viner et al. And this was from an autism paper. And essentially to evaluate common risk alleles in the trio sample, they tested for an overtransmission of polygenic risk scores. So this was done by simply looking at the polygenic risk score in the child, and then looking at the average polygenic risk score, the mid-parental polygenic risk score for both parents, and comparing them. So subtracting the mid-parental polygenic risk score from that in the child to calculate what they call the PTDT, so polygenic transmission disequilibrium test deviation statistic. So in a control sample, or in a control polygenic risk score, you don't expect to see any difference in polygenic risk between the proband and the mid-parental polygenic risk score. So you expect the p-TDT deviation to, on average, to be 0. But in a case scenario where you expect to see an overtransmission, you expect to see a positive p-TDT deviation, and we evaluate the significance of this using a t-test, seeing whether the p-TDT deviation across your sample significantly differs from 0. And in the autism study, they showed that yes, autism polygenic risk scores are indeed overtransmitted to autism probands. And what was quite interesting is that they showed there's a significant overtransmission of autism polygenic risk, even to those probands that carry a robustly associated high impact de novo variant. So we calculated polygenic risk scores in our new sample using independent training data. So PGC2 plus College UK for most of the trios, but for the Bulgarian trios that overlapped, we used PGC2 leave one out analyses. And we also looked at bipolar polygenic risk scores and, as a negative control, type 2 diabetes polygenic risk scores. So looking at all our trio samples, as expected, we saw a significant overtransmission of schizophrenia polygenic risk to the probands. Again, as expected, we saw a significant overtransmission of bipolar polygenic risk, but the type 2 negative control diabetes polygenic risk scores was completely flat. So we then wanted to look at the overtransmission of polygenic risk to our schizophrenia probands that carried a candidate de novo risk variant. And we defined this as de novos in either a loss of function de novo, in either loss of function intolerant gene, or in one of the known neurodevelopmental disorder genes. So in this slide, we can see in red, the degree of polygenic overtransmission to individuals that don't carry any of these de novo mutations. And then in blue, the polygenic overtransmission in carriers. So for example, if a synonymous de novo in a loss of function intolerant gene. So using that as a negative control, we can see that there's no difference in the degree of overtransmission between non-carriers and carriers of these de novos. When we looked at our candidate risk mutations, we saw a significant difference in the degree to which polygenic risk is overtransmitted, i.e. there is a significantly lower overtransmission in the probands that carry a deleterious de novo mutation. So this provides orthogonal evidence that, yes, indeed, these mutations are indeed contributing to the risk of schizophrenia and under a liability threshold model. So to conclude, we replicate previous enrichments of loss of function de novos and loss of function tolerant genes, and we show that this class of mutation are highly enriched amongst genes previously associated with neurodevelopmental disorders. We implicate SLC6A1 as a novel schizophrenia risk gene, and then we show that this polygenic risk scores are for schizophrenia overtransmitted to the schizophrenia probands, but we show that the degree of overtransmission of polygenic risk is significantly less in probands that carry a deleterious de novo mutation. So for future work, we're in the process of finding case control replication evidence for the SLC6A1 association. So just to acknowledge, sorry, that's okay. All our colleagues in Cardiff, all our collaborators, which have helped with the sample recruitment, and funding bodies. Thank you very much.
Excellent. Thank you very much, Elliott. So I'm getting concerned that the Q&A isn't working because there's no questions, which there should be. But if somebody can send me just a dummy response to say that there's anyone out there, I assume you're hearing us loud and clear. Okay, excellent. So Tammy's responded, so that people are listening. Okay, so thanks, Elliott. I think, you know, to convey what are quite difficult concepts in that amount of time is challenging, but I think you did it really, really well. So we should move on, and just to repeat that if people have got questions, then please ask them. Okay, so there's a question actually from somebody regarding that talk. So Richard Carlson-Lena has asked the question, Elliott, did you test for overtransmission of educational attainment alleles? So that's knowing a bit about this work, that isn't a simple answer, but
so yeah, we have been looking into other PRS, so educational attainment, and also IQ, and we, if memory serves, we did see a significant overtransmission of educational polygenic risk.
Yeah. And that's work that you're continuing to work on, and we're going to develop further, actually. Okay, excellent. I think we'll move on. But as I say, if people have got questions, then please ask them as we go, and then we'll hopefully have some time at the end in order to take that forward. Okay. So and next is Dr. Alex Richards. So I'll hand over to Alex shortly. So if we go to share. Okay, thanks. Oh, sorry.
Hi, I'm Alex. I work in the psychosis group here in Cardiff, and I'm talking to you today about polygenic risk scores and cognition in PGC2 schizophrenia cases. And this is work we've got just about to be published in Schizophrenia Bulletin. Okay, so there's a fairly well-known link between schizophrenia and cognition. In addition to the positive and negative and disorganized symptom domains in schizophrenia, you also get cognitive symptoms. In particular, schizophrenia patients have a lower IQ than healthy controls do. This has sometimes been found to be particularly true of performance IQ rather than verbal IQ. Lower premorbid IQ is also associated with a higher risk of schizophrenia, and cognitive impairment, the level of cognitive impairment of a schizophrenia case is quite associated with how well they do with their functional outcomes. And furthermore, antipsychotic medication, the main treatment we have for schizophrenia, does not improve cognitive impairment and may worsen it in many cases. So what we're doing today, what we're looking at in this study is based on the PGC2 schizophrenia 52 dataset, the meta-analysis of 52 schizophrenia datasets. This is mostly case control, but also contains trio data. All the subsamples we're looking at here are case control samples where we're just looking at the schizophrenia and schizoaffective cases. Some of the samples have cognitive data, subsamples of cognitive data, and we can use them. But the available tests and domains do cover quite a bit, as I'll get to in a minute. In addition to what was in the PGC2, we added some extra samples to the Cardiff COGS and Irish subsamples to maximize power. We also included the Work Package 2 UGI samples. This was a pan-European study from multiple European countries. And the Work Package 2 segment of it was a first episode psychosis sample where some of the samples became schizophrenic, and these are the ones we're including here. So the range in quantity and nature of the available cognitive tests that we've got presents a pretty major comparability problem. And the solution we use is to focus on G, the general intelligence factor. When multiple cognitive tests are performed on a sample, their results generally correlate to some extent. And this correlated portion of cognition can be calculated using a method like principal component analysis. And very importantly for us, it's generally robust to choice of tests or test domains. And we calculate G using multidimensional scaling rather than PCA because it can handle missing data. So our rule was when individuals had to have data from tests covering at least 2 cognitive domains, matrix domains, to be included. The UGI data was slightly different. It used WACE IQ, which had already been calculated, and had been calculated in a way that took into account the multi-country nature of the sample. It used multiple baseline data sets. Okay, now this slide sort of gives a flavour of the range of cognitive tests that we were dealing with. And I think the important thing to say is that although the tests do vary quite a bit, the domains that are covered are reasonably consistent. Nearly everyone's got speed of processing. Nearly every subsample's got working memory. Most of them have got, they've all got verbal learning and memory, and then some of them have attention and vigilance, and visual spatial learning and executive function tests too. And our total N, when we put all the samples together, is 3,034 schizophrenia and schizoaffective cases that have both genotype and phenotype data. Okay, so to begin with, we did a straightforward GWAS association between our individual genotypes and G using a mixed model analysis in loco in GCTA. This is a good analysis, takes into account population stratification and cryptid relatedness, and we meta-analysed the results together with metal, but unsurprisingly at this sample size, no result approached genome-wide significance. The nearest we got was on chromosome 2, but since none of these were within even an order of magnitude of genome-wide significance, we didn't explore these too much further. So the next step is to use polygenic risk score analysis to aggregate genotype, aggregate the effects of multiple SNPs across, and it gives you, this is, I think everybody here probably knows what the polygenic risk score is. It's a continuous additive measure of genetic risk of a phenotype for each individual. Which requires a training data set and a targeting data set, which are both got genotype information in it, and I cover the same set of SNPs and are independent. And the question we're asking here with polygenic risk scores is, how well do the PRS based on psychiatric disorders predict cognition in schizophrenia cases compared to polygenic risk scores based on measures of IQ in the general population? Okay. And so for these, we've got 5 separate training sets, 3 psychiatric disorders and 2 measures of cognition in the general population. This is fairly straightforward. The main thing to point out is that the schizophrenia data set obviously had to exclude samples that were in our cognitively informative target set. And so it's slightly smaller than the full PGC2 sample. It's also worth pointing out that we didn't include the COSAC2 samples here, as we didn't want to weight ourselves too heavily towards treatment-resistant samples, for reasons I'll mention just a little bit further on. We also had bipolar and major depression PRSs, and we had the most recent versions of the IQ GWAS and educational attainment GWASs to produce PRSs for IQ and educational attainment. And one thing to point out is that although the sample sizes vary quite a lot, in particular the educational attainment one is very large compared to the others, they do nonetheless explain reasonably similar, on the same sort of scale, amounts of variation in their own phenotypes. So it's not, there are not vast differences in power between these training sets. Okay, so standard PRS analysis. The PRS values are calculated within each subsample using PLINK, excluding the MHC and other regions of long-range LD. We use a p-value threshold of 0.05 for the primary analysis, as this was the threshold that explained the most various in the training set phenotypes. The only exception was major depression, where it was p of less than 0.5 explaining the most variance. Our results were generally very robust to this choice anyway. And the PRS are tested for association with G using linear regression in R, covariating for age, sex, and population principal components. And the results were then again meta-analysed across all the subsamples together using metal. And these are the results. They're pretty straightforward. Basically, we see a strong, a highly significant and effect with a reasonable effect size for education attainment and population IQ PRS. We don't see any significant relationship between PRS for schizophrenia, bipolar, or major depression and G in schizophrenia cases. We also calculated population IQ PRS in the biobank wave 2 samples. This had a somewhat higher effect size than the population IQ PRS did within the schizophrenia cases. And it's also worth pointing out this is not exactly the same population IQ PRS as was used in the schizophrenia cases, because it has to exclude the biobank samples that were present in Savage et al. So despite this, it still produces a higher effect size. So it does suggest that maybe, perhaps unsurprisingly, population IQ predicts best in a population sample. And just to zoom in on the results a little bit, and say that the results were pretty consistent. For IQ PRS, the results were pretty consistent across our different subsamples. I won't sort of dwell on it too long, but they all show the same direction of effect. And similarly, the schizophrenia PRSs are largely flat, apart from top 8, and so unsurprisingly, they lead to a flat meta-analysis. And lastly, yeah, the educational attainment one, again, apart from the Munich set, is again pretty consistent. Okay, we did perform a sensitivity analysis. This was just to exclude individuals who didn't have as many cognitive tests as the others. And we also left out the UGI subsample just to ensure that the different way of measuring cognition was make sure that wasn't affecting our results. Changed very little in our results. Okay, so to conclude, the PRSs based on population measures of cognition predict G in schizophrenia cases far better than, well, far more significantly than PRSs based on neuropsychiatric disorders, including schizophrenia itself. And this suggests that the mechanisms that underlie cognitive dysfunction in schizophrenia are at least partly independent from those that cause schizophrenia itself. And it's not hard to think of some possible categories that such variation could fall into. For example, genetic variation that alters the effects of antipsychotic medication, that makes you more susceptible to cognitive effects from that. Will produce this sort of result. And also if there's genetic variation that affects the disorder course subsequent to diagnosis, that may have an effect on cognitive dysfunction in schizophrenia without affecting the diagnosis itself. The other thing that this sort of tells us is that the well-known effects of schizophrenia genetic risk on cognition, they're well captured by the schizophrenia diagnosis. So schizophrenia genetic risk variants can have an effect up until the point where you have schizophrenia, but it doesn't appear that once you have schizophrenia, it doesn't appear that having more schizophrenia genetic risk worsens your cognitive outlook. One factor that could ameliorate our ability to find a link between schizophrenia genetic risk and cognition in cases is to do with rare variation, such as CMVs and de novo variants. Rare schizophrenia-related variation tends to adversely affect cognition and is believed to lower the common variation threshold that you need to become a schizophrenia case. And hence, this is going to oppose any positive correlation between common variant schizophrenia PRS and cognition in schizophrenia cases. Although it should be said that these variants are pretty rare even in schizophrenia cases, and so this may not be a massive effect. Thinking where we might take this next, we're thinking of looking at the effects of training sets enriched for treatment-resistant schizophrenia. Although, as I mentioned earlier, you want to be cautious when doing this because any polygenic risk score you base on a treatment-resistant set will become a marker for treatment-resistant schizophrenia, and treatment-resistant schizophrenics are known to have a worse cognitive outlook than non-treatment-resistant ones. So therefore, it would be interesting to try it both within treatment-resistant categories and across. Naturally, at some point, we'll try higher-powered training sets like the PGC schizophrenia, and potentially we could look at subdividing cognition up into tests or domains, although the main issue there will be maintaining sample size and power while being able to perform these tests. And we may also examine polygenic risk scores for other disorders, particularly, cognitively relevant ones like autism, developmental delay, and intellectual disability. Okay, well, thank you very much for listening, and thank you to Mick, Mike, and James for overseeing this work. Thank you to Catherine, Antonio, and Aura who started this work off before I took over it, and thank you to everybody involved in the PGC and UGI consortiums.
Because various postdocs have been doing it in non-dedicated time, but it's really good to see it come over the line, and hopefully this will serve as an impetus for wider groups and additional people to contribute data and get involved in these kind of analysis. Because we're just for the audience to know that the PGC group more widely are beginning to consider what goes into the next grant, and one thing that we're seriously thinking about is the the value and possibility of working more with phenotype data, both within and cross disorders. It does present a lot of challenges in data amalgamation, but I think efforts like this and those that the cross disorder group have done, that Rudifer and others have done, looking across schizophrenia and bipolar disorder, are really good contributions to overcoming those hurdles. And it's something we certainly need to do if we're going to get out of the diagnostic category boxes that we know are imperfect. Okay, so let's just have a look if we've got any more Q&As.
Chat better.
Okay, so there's a question for Sophie from Richard Josiason. Wonderful presentation, I thought that as well. One of the difficulties of presenting to a blank screen is the lack of any feedback. So I was going to say, if you know, I'm sure you want to applaud the postdocs, but I'll do that as well, just to give them some degree of feedback. But thanks, Richard, because it's nice to have some feedback. So Richard asked, what were the reasons for selecting individuals with psychotic experiences? What were you expecting to find, maybe thinking they're at higher risk for full-blown psychosis? So why did you ask the question, I suppose?
Yeah, so the epidemiological work indicates that these people are at higher risk of mental health conditions, but they're so much more common. And really a lot of the work that I've been doing is not thinking so clearly about disorders as categories and looking at more endophenotypes, and I'm wondering whether that will help to give us more insights into disorders than we have at the time. So we went into it thinking that there might be something more specific with schizophrenia, but there's not. There's really not. So it was quite interesting to see that it was so unspecific. But that was the motivation, really, is just trying to think of other kind of phenotypes that we can look at that might help dissect the heterogeneity of schizophrenia and related disorders.
And I think as well, it's worth noting that within the clinical context, then psychotic experiences are seen as being something that can be an augur of transition to full-blown psychosis. But I think a lot of us are doubtful about their ability to do that. And this actually reinforces that idea that psychotic experiences, in as much as they're captured within UK Biobank by self-report, probably are no better predictor of psychosis. Well, the genetics underlying schizophrenia is no more correlated with the genetics of psychotic experiences than is the genetics of depression, etc.
And I think, as you've just gone on to say, actually, that there have been some really great epidemiology studies already of psychotic experience. And it's really interesting, you said the mostly developed depression. And actually what we find is that the genetic correlation and the polygenic risk scores were certainly higher for depression, much higher than they are for schizophrenia. And Stan Samet, who is also supervised this work, suggested that it may be because the psychotic experiences seem to co-occur with some mood sort of change, but I think it's quite interesting, to see what's causing them. Yeah,
and as Richard has rightly pointed out, then the epidemiological research itself indicates that those who are in so-called at-risk mental states or are at risk of developing psychosis in adolescence and early adulthood, it's a small percentage of those that go on to get schizophrenia. So it's a poor predictor in that respect, and more go on to get more common conditions like depression and anxiety. Okay, thanks. I don't think there are any more questions there, and then there are no additional Q&As. So I think at that point we'll wrap up. I just want to thank Sophie, Elliott and Alex for doing a sterling job this afternoon. So thank you very much, and I'm sure you're happy for people to contact via email if they have further questions, and certainly we'll be keen to expand on this work as part of the PGC as well, in the schizophrenia group and more widely, I'm sure. The other thing I just wanted to highlight was that this is a newish initiative to encourage early career researchers to present their work in this forum, and it's something that we'd really like to encourage. So if there are other researchers at that stage that would want to contribute, then we're going to hopefully host something like this every 2 to 3 months, and so we could bring people together. It doesn't have to be from one centre, it can be across centres, and it's a way of presenting and hopefully discussing and getting good input into the research that might be at the stage of submission or early publication. Thanks very much everyone for joining us this afternoon, and enjoy your weekend. Goodbye from sunny, I would say sunny Cardiff, it's not sunny at all. Sunny Cardiff in Wales, which is spelled W-A-L-E-S, and isn't the massive mammal that Donald Trump thought it was. Thanks everyone, all the best. Bye.